Hi, good morning and good evening to all of you. I am Wendy Kleiberg, EU QPPV. Welcome to the webinar today. The topic for today's webinar is Pharmacovigilance and Medical Monitoring on a Global Platform, Overcoming the Tribulations. Well, in-depth knowledge and profound discernment are mandatory for satisfactory clinical safety and medical monitoring services. The tricky situations of this arena often leave the safety professionals exasperated. To comprehend the nuances of the field, we thought to come up with this webinar on pharmacovigilance and medical monitoring on a global platform, overcoming the tribulations. Before I introduce our speakers, I would like to cover a few housekeeping topics. Today's webinar is being presented by Awinsa Life Sciences, a global provider of pharmacovigilance, clinical safety, medical information, and medical writing services. Manned by people with discernment and an eye for quality, we at Awinsa ensure astute analysis of safety reports so that clinical scenarios emerge in perspicuity. You're welcome to post your questions during the webinar. We will do our level best to address your questions. In case we are not able to answer your questions due to time constraints, we will try to respond to you via email. The webinar will be available for download on demand after a couple of days. Today's presenters are Dr. Sanjeev Miglani and Dr. Mugdha Chopra. I will now introduce our speakers to you. Dr. Sanjeev Miglani is the founder and director of Awinsa Life Sciences. He is an MD in internal medicine and has over 20 years of experience in the field of medicine, pharmacovigilance, and clinical research. In his last assignment, he was the Vice President of Safety and Medical Affairs at EPSA. Just before joining EPSA, Sanjeev was the Vice President for Pharmacovigilance and Medical Writing at Accenture and had been in senior leadership roles in some other companies like CIDP, CliniRx, and Ranbaxy prior to his stint in Accenture. He also worked as a senior resident in cardiology and medicine department. Dr. Mugdha Chopra is the co-founder and director of Awinsa Life Sciences and has over 16 years of experience in pharmacovigilance and clinical research. She was Associate Vice President for US PV and Clinical Safety at EPSA in her last role and was overseeing the delivery and operations from India, which included case processing, aggregate reporting, literature monitoring, and signal detection. Before joining EPSA, she was with Ranbaxy as a compliance lead. She comes with an extensive experience in various safety databases like Argus Safety and RSG, and has also met the challenges of business continuity planning and data restoration. At this time, I'm going to hand the floor over to Dr. Sanjeev Miglani, who is now going to start today's presentation.
Thanks, Wendy, for such a nice introduction. So today, good morning, good evening, everyone. So in today's webinar, we're going to talk about the clinical safety management and medical monitoring in the global clinical trials. And we're going to cover the following topics today. We're going to talk about the management of serious adverse events in the clinical trials, how we do the signal management. I'll then touch base upon medical monitoring aspects of clinical trials and some of the common examples or issues that we observe at the site level. My colleague Dr. Mugdha will then talk about the clinical trial safety reporting requirements and how the follow-up should be done in the clinical trial pharmacovigilance process. She'll also talk about auxiliary medicinal products, and at the end she'll talk about the challenges in clinical safety. All right, so to begin with, I'll briefly talk about the process that we have to follow in the clinical trial of pharmacovigilance management. So at the site level, and I don't need to talk about the basic terminologies. I hope the audience is well versed with the terminologies like adverse events or serious adverse events or the reportable cases, which are SUSARs. So to begin with, the first and the foremost thing that happens is at the site level, when the site has to report any adverse event or serious adverse events. The sites normally do that on a CRF, which is generally electronic CRF nowadays by and large, and the sites would capture that adverse event or SAE in the ECRF, and accordingly then the information gets reported to the safety team. Of course, when there is a SAE which is reported by the site, so as a part of the process, either the sites will enter that SAE information in a kind of a dedicated SAE form, which could be a manual form, and sites then send that SAE form to the safety team at their dedicated email ID or their dedicated fax number, and that's how the safety team gets the information. Or sometimes it can be purely electronic process where first an email alert is generated, and then from the EDC system itself, a PDF output or any kind of a typical safety report can be generated by the electronic database system or the EDC, and through that particular EDC system, the safety report can be routed to the safety team who does the case processing of the SAEs. And if it is just a adverse event, which is not a serious one, so that remains only in the CRF, and when the medical monitors are reviewing the data in the CRF, they can identify any discrepancies or error in the monitoring process. So this is just a kind of a high level idea that I'm giving about the overall management of the SAEs in the clinical trial space. So the moment the E2B transfer is done, or the SAE E2B transfer is done to the safety database, either through the electronic process, or if the SAE is sent by some email, the safety team gets the information, and they typically would process the case. So there is a team who does the data entry, followed by QC and then the medical review. And at the end of the day, the safety report in the form of SIOMS or MedWatch or any E2B report, if required by any regulatory authority like FDA requires that, or like EMA needs the E2B report. So the submission has to be done. And while the case processing is done during this process, if there are any queries that are required to be sent, so that is typically done by the safety team, and the queries are sent to the sites. Again, the process can be either through EDC directly, wherein the queries can be posted on the EDC and the site can just respond through the EDC, or it can be a query form wherein the sites are responding to the queries sent by the safety team. So this is just a high level process map which basically talks about the pharmacovigilance process for the SAEs which are processed in any Argus database or any safety database for that matter. Now, as far as the reporting obligation of the investigator is concerned, it starts from the time when the patient signs the informed consent, and it is applicable till the end of the follow-up period, which is typically till the 30 days after the last administration of the study drug. Now, when I say that the obligation is there up till 30 days, this is generally a timeline which is more or less followed in most of the protocol, but sometimes this timeline can vary, and typically it would depend on the half-life of the product. Sometimes the half-life of the product may be much longer, or sometimes the drug stays in the body for a longer duration depending on the PK parameters. So in those scenarios where it is expected that the drug will remain in the body for a longer duration, so there even the follow-up could be expected even up till 60 days also, and those could be some uncommon instances. And the second thing is that if they are expecting a particular adverse event that happens even say beyond 30 days or beyond 60 days, but we think that there is a drug relationship or an event relationship with the drug, then those kind of events have still to be reported. So practically any event where we have a strong suspicion of causality, irrespective of duration, it has to be reported even beyond this particular time frame. Say for example, there is a patient who develops carcinogenicity or say hepatotoxicity, and there is a suspicion of drug-induced etiology. So in those cases, the follow-up has to be done even beyond that particular time period. And every attempt should be made by the investigator to follow up with the subject until the adverse event is resolved, or until the investigator determines that the subject has returned to the acceptable state of health. A very important aspect, I know many of you would already be aware of the causality assessment basic process, but first of all, we all need to understand that causality assessment means that the likelihood of association of the event with the drug, and there are a lot of factors that have to be taken into account when we're doing causality assessment, and it has to be done at both the levels. The investigators have to provide the causality assessment at their level, but when the SE form comes to the safety team. So the safety physicians have to do the causality assessment based on the clinical presentation, based on the parameters which are provided by the site. And the factors that have to be taken into account include the temporal association between the study drug and the adverse event, the subject's underlying clinical condition, because sometimes it could be the underlying condition that is causing the SA. Sometimes it could be the concurrent or the concomitant medication which might be causing the adverse event or the serious adverse event. And then, of course, we have to take into account various factors that can actually help in making a kind of a interpretation whether this event is related or not. Say, for example, if we get to know that the D challenge was positive. When I say D challenge, I'm trying to say that the event is basically subsiding after the drug is discontinued, or say when the event is when the drug is given again, the event again starts. So that's the D challenge. So if something like a D challenge or D challenge is positive, so that would indicate that there is a strong causal association, and the causality would be in the favor of drug. So broadly, uh, many people in the industry use the WHO classification, which says definite, probable, possible. So all of these get classified under related. And then if you have something like unlikely or not related, so that's typically a classification of causality which is not related to drug. Practically, if we look into the regulations, whether it is from EU perspective or whether it is from the FDA perspective, or most of the regulatory authorities globally, they would need the causality as either you are saying yes or no, either you are saying it is related or it is not related. Because even though we may classify that a definite, probable, possible may come under as related, at the end of the day, decision has to be taken whether it is a SUSAR or it is not a SUSAR. SUSAR means suspected, unexpected, adverse reaction, which is the event which is related to the drug. And if it is a SUSAR, it means that we are saying that there is a relationship with the drug. And overall, if we go specifically, sometimes the protocols will define what is definite causality. By definite causality, we're trying to say that yes, event is definitely related to the drug. When we say there is a probable causal association, we mean to say, and WHO defines that it is more likely to be due to drug and less likely to be due to other factors like the concurrent medications or the concurrent illness or the underlying medical history. Similarly, when we say possible, it means that it could be either way around. We don't have much information, and the causality could be either way around. It could be probably because of the underlying disease or the conditions, or it could be possibly due to other factors or to the drug. Similarly, when we say unlikely, unlikely means that it is more likely to be due to other factors and less likely to be due to the study drug. So that's how the WHO defines the causality assessment. So as such, there is no gold standard for doing causality assessment. The only thing is that in the protocol, we have to define clearly what exactly would be taken as related and what would be taken as not related. I would like to now draw your attention to the FDA rule that came in 2012, which was basically IND safety reporting rule. And that particular regulation or rule that came in 2012, it provided a clarity on the concept of reasonable possibility. Now, before that, there was a kind of a ambiguity, or we can say that the process was not very clear. There was a lot of over reporting done by the sites. The sites used to take conservative approach. So FDA tried to basically come up with this guidance document in 2012, and came up with this anti-safety reporting rule, where FDA provided a lot of clarity, like what kind of events should be considered as related, and what is the concept of reasonable possibility when the concept of causality has to be used for the clinical trials. So FDA gave three scenarios wherein the clarity on the causality assessment was given. The first point was that if there is a single occurrence of an uncommon event, then the causality should be considered. And if we know that a particular event is known to happen with that kind of with a drug, then those events should be reported as a SUSAR if it is unexpected and if it is related. Let me take some example. Some of the events are very commonly known to happen with a drug, like say agranulocytosis or hepatitis or drug-induced liver injury, or myelosuppression, or we can say rhabdomyolysis. So these kind of events are typically known to happen with the drugs, and if there is a particular adverse event that happens after the intake of a drug or a study drug, then they should be considered kind of unexpected. And of course, expectedness will be depending on the IB, and if the event is mentioned in the IB, then it should be unexpected. But if such an unexpected event is happening because of a particular drug, and these typical events which we know are typically drug induced, so they should be considered as related to the study drug. Now, the second scenario of the reasonable possibility which FDA gave was if there are any one or more events which are not commonly known to happen with the drug exposure, but if there is a temporal association, and if we know that yes, some de-challenge or re-challenge is also occurring in those kind of patients, so they should also be considered to be related. So depending on the clinical presentation, this decision has to be taken. So and in fact, many cases or events in the past have been found based on this kind of scenario, like tendinitis in drugs which are from fluoroquinolone groups. Um, so in such kind of cases, you can see that there is a temporal association, and uh, in these kind of scenarios, we can see a typical de-challenge phenomena. The events would subside typically on discontinuation of study drugs, and we can see that the re-challenge is also positive in these kind of cases. And the third scenario is that when there is an aggregate analysis of specific event observed in clinical trial, and after doing this aggregate analysis, if we come to know that a particular adverse event is happening more in a particular age group, then if we find such particular association in a particular age group compared to placebo, or in a particular defined group other than placebo, then we can report those cases if we get to know that yes, there is an association. In fact, FDA gives clarity on this kind of scenario. Earlier, sometime when in the clinical trials, the cases of say myocardial infarction or strokes, they were reported based on the conservative approach. They were reported as SUSARs, but generally these kind of events, say typically if they're happening more in the elderly population, uh, they are more likely to be due to the underlying condition of the patient rather than due to drug, because we don't expect some event like myocardial infarction or stroke to happen with the study drug. I'm not saying that these kind of events cannot happen with the study drug. We know obviously there are a lot of drugs which can cause MI or thrombotic events, or events like stroke, like roxacoxib, which came in, it was causing myocardial infarction or thrombotic events. So there are a lot of drugs which can cause thrombotic complication. But the point that FDA tried to basically clarify was that generally these kind of events would not typically happen with the study drugs or the drugs. So we should always kind of apply our medical judgment, and only if we are finding a particular aggregate analysis, and that particular analysis is indicating that yes, the event is happening say more in the studied drug arm compared to the placebo arm, then those events should be reported, and then the causality again should be considered to be related to the study drug. I'll now talk about the analysis of similar events. So this basically reporting of analysis of similar events is a classical requirement of the US FDA. Not many regulatory authorities require AOSE. Like in Europe, there is no requirement for providing a report of analysis of similar event when a SUSAR has to be reported. But in the US, it is a clear mandate from the US FDA that whenever there is a 7-day alert report or there is a 15-day alert report, a report which is called analysis of similar events should be given along with the E2B report. So as a part of routine practice, what most of the companies they do typically, they would prepare a summary of analysis of similar event, and they would put that summary within the safety report that is submitted to FDA, which is acceptable. Now, what happens in this particular process is, first of all, we have to identify the similar kind of events that were reported when a SUSAR is reported, and identification can be done through different ways. Either it can be done manually by looking for the similar preferred terms within the Medra browser, or sometimes we use SMQs. When I say SMQ, I mean to say a standardized Medra query. And a standardized Medra query means that first of all we pick up that standard meta query from the Meta browser. Say if we have a case of allergic reaction, and in SMQ of in a Meta browser there is an SMQ for hypersensitivity, so we can identify all the broad level terms from that SMQ of hypersensitivity from Meta browser, and then what we do is we identify all the cases which are having all those terms which are a part of hypersensitivity. So by this, it will enable us to identify all the cases in the safety database which contains or have any kind of events pertaining to hypersensitivity. So that is the first part, which is called identification. And then there is the analysis which has to be done for that data. So I'll show you in the next diagram how this process can be simply followed. So you see in the bottom part, once we get the assay, we apply a SMQ search, which is like identification of the cases which have the terms of that SMQ. And you will have sometimes challenges. You might not get the exact SMQ. So like I was giving the example of allergic reaction. So you might have an SMQ of hypersensitivity in Medra, but probably you might not be having SMQs for everything that you're trying to see. So you might be having some scenarios where you might be just picking up some preferred terms, and you are trying to then dig the data or the cases from the database which are containing those particular adverse events. Now I'll give another example. Say if we are dealing with a SUSAR which is having an event of myocardial infarction, so maybe we'll have to look for different kind of terms, or maybe we'll look for some SMQ. But then our idea is that similar kind of events like say angina or subendocardial infarction or coronary artery disease. So any event which is having ischemic cardiac event, so they should be extracted from the safety database, and then we have to generate a line listing so that we can exactly derive all the cases which are having the terms similar to the SUSAR, and then we do some analysis to see if there is some meaningful interpretation that we can do for the data. So at the end of the day, when FDA is expecting analysis of similar event, FDA is expecting that the sponsors would try to make a meaningful clinical picture of that particular adverse event and see if a particular association is coming up with the study drug or not. So and that's why the concept is to either use SMQs or similar PTs, but dig out all the cases with that particular term and see if there is a study drug association with that event. So that's the basic concept. The next part I would like to cover is the signal management in the clinical trials. And generally, the signal management in clinical trials is not largely different from the post-marketing scenario. The only thing is that in the clinical trials, we don't get as many cases that we normally get in the post-marketing. So we do apply statistical methodologies also like chi-square or PRR. But for clinical trials, if the number of cases are very few, or especially if we are focusing more on the assays, then we have to rather focus more on the qualitative approach, where we actually try to assess the quality of the assays, the content, and the information that is provided in each and every assay. Because anyway, in the clinical trial, it's a very controlled environment where there are limited patients with limited assay data. So when we are anyway focusing on the assays, especially when we are concentrating on the event which are related, so if the volume is not very high, our intent is to look into the actual clinical picture for each and every case based on the clinical presentation of the patient, and then we try to make a meaningful clinical picture. So of course, that is always a very important methodology for clinical trial cases when we do the signaling. But yes, if the data is more, especially if it's not a double-blinded study, if it is a open-label study where we exactly know if there is a adverse event data for the study drug, and then you have the adverse event data for placebo, then of course we can always use the statistical methodologies also, and then make a kind of a judgment whether the events are related to the study drug or if they are not related to study drug. And depending on the signal or depending on the data that we get after doing the signaling, different kind of decisions can be taken. Sometime the IB can be amended. If you look at the CTFG guideline, they typically mention the criteria how to update the IB based on the signaling that we do. And recently, when CTFG came up with the new guidance, it mentioned that for any update in the IB, especially in the RSA section for the SAR, so there should be at least more than one serious adverse reaction that has been reported, then it should be captured in the RSI. And there can be some exceptions to the rule. Sometimes there can be just one SAR also, but if we know that there is a strong causal association, when I say strong causal association, I mean to say if there is a positive D challenge and a positive re challenge, or if we have enough evidence to indicate that yes, there is a clear-cut rationale of ruling out the other factors, and yes, we know that there is a clear-cut association with the study drug. So in those kind of scenarios, even if there is one SAR or one serious adverse reaction, RSI can be updated to include that. Otherwise, CTFG guideline very clearly mentions that unless you have more than one SAR, they should not be included in the RSI section, which was, and that's what CTFG came up in their recent guideline. The other thing is that if there is a new signal that might require any amendment in the protocol, maybe like if we get to know that, okay, this drug might have a hepatotoxic potential, so there might be some changes in the inclusion criteria. There might be some scenarios wherein we might need to, of course, change the IP, but in the DSUR, we have to communicate to the regulatory authority and mention those new safety concerns. So depending on the kind of data that we get, the decisions have to be taken accordingly. I'll now focus on the medical monitoring part, and in the medical monitoring, maybe I'll give some examples of the common issues that we normally identify. But to begin with, I'll just first talk about when exactly medical monitoring starts. Well, it starts even before the study starts, because the medical monitors have a role to play in the review protocol. They play a very vital role in the training for the sites or the SAs, the management of the SAs. They provide the training in the investigator meetings or the site initiation visits, or they resolve the queries coming from any ethics committee or from the sites. And now when the study actually starts, there are a lot of activities where medical monitors get involved in. They would be involved in patient eligibility. So there could be a specific eligibility form, and the medical monitors have to review those forms and then let the site know whether the patient is eligible or not. Then there could be data points pertaining to lab or ECG or efficacy listings or safety listings. So the medical monitor has to regularly review those listings, and in the medical monitoring plan, so it should be defined whether those listings have to be reviewed on a monthly basis or on a bi-weekly basis. And then if there are any queries which are identified, generally the queries can be reviewed, or the data can be reviewed in the form of line listing, or the medical monitors can directly go to the EDC and then review the data. And if they identify any queries, they can post those queries on the EDC directly. I'll show you some examples, like the common kind of errors. And these are hypothetical examples that I will show you. And of course, when we close the study, medical monitors are involved in the review of the CSRs. They would identify any particular safety concerns if they are evaluating the data. So that can happen either during the time of the study conduct, or maybe after the study gets closed, when they are evaluating the overall safety data. Now, I just draw your attention to some of the common kind of errors. Now, again, these are not some actual examples or the site cases. I'm just trying to show you some hypothetical examples. These kind of things can happen at the site level, and if the medical monitors identify these kind of issues or errors, so medical monitors should actively discuss these kind of scenarios with the investigator and discuss that, so that the data is clean in the clinical database. Now, if you look at this example, you can see that there are different kind of ECG data for PV wave, PR interval, but there is a clear ambiguity in the QRS. We know that the QRS duration is typically between 80 to 100 or 120 millisecond, but this is a clearly abnormal QRS duration. So if the investigator fails to document the clinical significance, or if he or she does not mention that yes, this is abnormal and it could be indicative of some particular adverse event, so then the medical monitors should at least discuss that. Maybe there is a rationale by the investigator, but these kind of things should be discussed actively by the medical monitor. Now, in this particular example, you can see that in this last patient, the QTC change from the baseline was more than 60, and FDA clearly mentioned that if there is a value absolute value which is more than 500, it becomes clinically significant. Or if you have a QTC change from the baseline which is more than 60, so it should be evaluated because it could be clinically significant. So whenever we encounter such data where the change from the baseline is significant, so the investigator should document the clinical significance. Maybe sometimes there could be some scenarios when the data may not be abnormal, but it is very important that there should be clarity on the documentation. And if there is such kind of data, so the investigator should provide the clinical significance, and the medical monitor should always discuss that, so that tomorrow those questions may not come up at a later stage. Now, this is an example where we have some lab data. We can clearly see that AST is around 3.5 times the upper limit of normal. ALT is also around 2 times the upper limit of normal. So had it been just one enzyme, maybe which is, say, very slightly elevated, it may not have that kind of clinical significance. But in this scenario, there are 2 enzymes. So generally, when we are having 2 hepatic enzymes which are elevated, of course, you would like to see other things also like INR or bilirubin. But then this cannot be just a coincidental finding. So the medical monitor should look into it actively. Yeah, maybe if there is a single hepatic enzyme which is, say, just around 1 or 2 times, sometimes just because of exercise, because of hypoxia, the liver enzyme may raise, and then it will come down to normal. So that can be explained, but sometimes the clinical picture cannot be explained, like in this example where there are 2 hepatic enzymes which are having an elevation of 2 to 3 times or more than 3 times the upper limit of normal, like for AST. So these sort of things should be very closely monitored. Now, again, in this example, you can see sometimes the lab data itself could be not as you can see that in this case creatinine is 125, which is not practically possible. So obviously nobody can survive at this value. So clearly it is a lab error. So at times there could be even lab error, and then in those cases investigator has to medical monitor has to anyway dig that data, and then has to check with the lab, and then lab can give a clarity, because this data is not possible at all. So maybe the lab was trying to give 1.25 or something, but 125 is not something which is practically possible. Now, this is an example of an oncology trial where the efficacy is evaluated based on the single largest dimension. So the single largest dimension for the solid tumors is measured, and based on the reduction in the size of the tumor, the efficacy is determined. And if the patient is having improvement in the single largest dimension, so the patient may be classified as partial response or complete response. In this example, you can see that patient was showing improvement, and the single largest dimension was declining gradually, and the patient was going from stable disease to partial response. But then all of a sudden, the value of single largest dimension started increasing. But then you can still see that the patient is being classified as partial response. So maybe these kind of things can be discussed with the investigator to get clarity whether this data is actually correct, or maybe somewhere the information has not been captured correctly. Now, there could be some graphical representation of the data also. It's not necessary that the medical monitors have to review the line listing or have to review the EDC. Based on the data that is derived, or based on some of the graphical representation, medical monitors have to review that data also. Now, this is a classical example of a box and whisker plot, and this particular plot in this scenario represents a variation in the total hemoglobin concentration of different grades of liver fibrosis, and the middle line represents the median. The bottom and the top of the box is 25th and 75th percentile of the data, and the line extending from the top and bottom of the box represents the upper and the lower extremities. Now, in the next example that you see, the scatter plot, so we can see that there is a significant positive correlation which was found between the maternal and fetal hemoglobin concentration, and this was typically just seen between hemoglobin of the mother and the fetus, but the similar correlation was not found between the maternal and fetal WBC, RBC, and the platelet count. I'll briefly talk about the Hy's laws in some investigation products. Hy's laws is very important. In the Hy's laws cases, what is more important is that we have to see the value of AST and ALT and the alkaline phosphatase. And if AST or ALT is more than 3 times the upper limit of normal, and the bilirubin is more than 2 times the upper limit of normal, so that is a positive Hy's laws. And if only AST or ALT is 3 times the upper limit of normal, but the bilirubin is less than 2 times the upper limit of normal, so that is just a Temple's corollary. It's just a kind of a indication of a liver injury. And when we see this kind of pattern, it is indicative of a drug-induced hepatocellular damage. And obviously, when alkaline phosphatase is low, it indicates that your obstructive pathology is not there. Like you're not having any kind of obstructive jaundice or any obstruction of bile ducts, but it is more in favor of the hepatic or hepatocellular damage, and which is more of drug-induced. And then there can be percentage change from the baseline, and this kind of data when we look at the graph. So this can be used both for efficacy as well as safety parameters. This is just one example wherein, though again, as I mentioned, these are just hypothetical examples, but at times the site may just miss that patient actually got hospitalized and there was some action taken, but the assay was not reported. So the site should be very careful that if patient gets hospitalized, it's a very standard criteria. So the case has to be captured as an assay if the hospitalization happens. All right, so I think I'll now hand over the session to Dr. Mukta. So she'll talk about the other topics. Over to Mukta.
Yeah, thanks, Sanjeev, for sharing the details about clinical safety and medical monitoring aspects and the associated challenges. Modern era of pharmacovigilance starts with an intent to capture sufficient evidence of safety and efficacy. And clinical trials lay this foundation to capture the safety data emerging from the clinical trials. And safety reporting is one of the important and critical attributes of clinical safety. So today I would be talking about safety reporting requirements in clinical trials for some specific countries. Other critical topics in clinical safety reporting like management of follow-ups, unblinding, and safety assessment committee. Before I talk about safety reporting requirements, I would like to highlight the importance of pharmacovigilance in clinical trials. And why is safety reporting important in clinical trials? Safety invariably gets captured through pharmacovigilance, and first and foremost significance of safety reporting is that regulatory agencies are becoming proactive in seeking out potential safety issues. Of course, there is an ethical requirement to report adverse drug reactions coming out of clinical trials. And last but not the least, non-compliance in safety reporting can lead to warnings, penalties, and even closure of the companies. So most of the pharmaceutical companies are on their toes to maintain compliance to the global safety reporting requirements. This is a high-level snapshot of how to carry out the safety management in the clinical trials. So safety management process is same for both clinical trials and post-marketing scenarios, which includes collection, collation, analysis, and communication to stakeholders. For clinical trials, if you see on the extreme left, it is the clinical trial data which is collected, and then the data for serious adverse events is processed systematically into the safety database. Further analysis is done for this processed data to identify any signals, and this data is accordingly then communicated to different stakeholders, and communication involves submission of relevant issues to regulatory authorities as per the submission requirements, investigator brochure updates based on the data analysis, and any responses to be provided to regulatory authorities for any inquiries for safety of the product. Now, before I touch upon the safety reporting requirements, it is very important for all of us to understand the basic terminologies in pharmacovigilance, and all of us are well aware of that. So these are the adverse events and serious adverse drug reactions, and SUSARs. So there are serious adverse events, and amongst them are the serious adverse reactions, which are serious and related, but these are expected as per the reference safety information in the IB. SUSARs are suspected, unexpected, serious adverse reactions and are the ones which are considered for reporting by and large many authorities. However, there are some authorities like China who actually which require the serious adverse reactions, drug reactions to be reported to them and not just SUSARs. So requirements are laid down by the regulations, and it is very important for us to interpret the regulations appropriately to meet the requirements. So if you see this is a glimpse of the reporting requirements at a very high level for some of the countries. And I'll definitely go into the specific details in my subsequent slides. But at a broader level, if we see, most of the regulatory authorities require expedited reports, as everyone is trying to move towards ICH GCP from the perspective of safety reporting in clinical trials. Aggregate reports are also now required by most authorities, barring a few exceptions if we see here, like Health Canada, where the aggregate reports are not mandatory for submission and they are just submitted on a voluntary basis. And for Australia, where TGA does not require the aggregate report to be submitted to them, but in fact it is submitted to the ethics committee, which is Human Research Ethics Committee for TGA. I'll now talk about the country-specific requirements, first one being the US. So US FDA requires SUSARs to be submitted to them. Fatal and life-threatening SUSARs are submitted in 7 calendar days and rest all other SUSARs in 15 calendar days. A very specific and interesting fact about reporting to US FDA is that sponsor assessment here takes precedence over the investigator assessment. So now what does it exactly mean? It means that even if investigator considers any event as related, however, if sponsor considers it to be not related, it is not required to be reported to FDA as a SUSAR. The detailed assessment and documentation has to be captured by the sponsor in the safety reports. And another important fact is the submission of analysis of similar events with SUSARs, which Sanjeev explained in detail in his slides as well. So this is generally a specific requirement for submission of SUSARs to FDA and generally not required by the other regulatory authorities. US FDA is also coming up with a new guidance on electronic submission of SUSARs through the electronic submissions gateway to FDA. FDA has already published a guidance for electronic submission of IND safety reports, which provides the details of which all reports will be accepted by FDA electronically in XML format from a clinical trial perspective moving forward. And it will obviously bring ease to the sponsors. The exact date has not been published by the FDA as of now as to when FDA will start accepting IND safety reports in electronic format, but this will be a new step towards harmonization in terms of information received by FDA in their database. With regard to the aggregate reports, DSUR or IND annual reports are required to be submitted to FDA on an annual basis. Similarly, Canada also has the same reporting requirements as of the US, that is SUSARs in 7 calendar days for fatal life-threatening and 15 calendar days for all other SUSARs, but they have to be reported to Health Canada via fax. In Canada, the DSUR submission to Health Canada is voluntary, not mandatory, as it is in the US or some of the other specific countries. In Europe, there are many countries, but obviously I'll not be talking specifically about the individual countries in the session right now. But by and large, the requirements in terms of timelines of submissions are standard across Europe. There are still some country-specific requirements in terms of modality and format of submission of the reports. In Europe also, SUSARs are required to be submitted, but unlike FDA, where sponsors assessment takes precedence in FDA, in US, in Europe, if either investigator or sponsor considers the SAE as related to the investigational medicinal product, it has to be submitted as a SUSAR. In Europe, there can be central ethics committees, there can be local ethics committees where submissions are required to be done for the SUSARs. Ethics committees across Europe have different modes of submissions. Some ethics committees accept the IND safety reports via email, but at the same time, there are certain ethics committees which have specific portals through which the submission is required to be performed. Like Austria has a specific portal, Netherlands has a specific portal, CMO portal, where the submissions of the IND safety reports have to be done. Europe also requires DSURs to be submitted to the competent authorities on an annual basis. The submission process is being harmonized as a common European submission portal, that is CESP, has been developed, and submissions are being done through CESP portal for certain countries in Europe. This is another move towards harmonization of the requirements. A new regulation, which is Clinical Trial Regulation 536/2014, is coming up for harmonizing the requirements across Europe. This regulation actually entered into force in 2014 June, and the timing of launch completely depends on the European clinical trial portals and databases. So EMA is planning for January 2022 for the go live date of EU clinical trial information system database and portals post the audit of the systems. And once the portals and databases are fully functional, clinical trial regulation will come into effect. Talking about Japan, the expedited reporting and aggregate reporting requirements are similar to the ones in US and Europe. However, there is a striking difference, which is that expected death cases are also required to be reported to PMDA. And if we talk about South Africa, the regulatory authority of South Africa is SAPRA. And SAPRA requires the expedited reports to be submitted to them, but here the timelines are different from some of the other countries, which we talked about earlier. In South Africa, domestic, fatal, and life-threatening SUSARs have to be reported in 7 calendar days. All other local SUSARs have to be reported in 15 calendar days. But if we talk about foreign, fatal, and life-threatening SUSARs, they have to be reported in 30 calendar days to SAPRA. And follow-ups of any such foreign SUSARs can be reported on a 6-monthly basis. With regard to the aggregate reports, DSURs have to be submitted on an annual basis. But apart from the DSUR, there is a specific requirement for SAPRA, which is a 6-monthly progress report, which also has to be submitted. The next country is Mexico, and the reason why I've taken Mexico is that there are some specific nuances in the reporting requirements for Mexico. Apart from the SUSAR reporting, there are certain other reports which are required to be submitted to the authorities. If there are 2 or more serious cases in the same place with the same drug of the same batch, they have to be submitted within 48 hours. Also, lack of efficacy cases have to be submitted in 15 calendar days. And apart from all this, safety issues from scientific literature also has to be reported within 30 calendar days to Mexican authority. These requirements generally are also applicable for post-marketing, but Mexican authority has given a clear message that these submissions will also be done, are required to be done for clinical trials as well. So the submissions have to be done via CNFV website, and investigators have to be made aware of the reports via email. And talking about the aggregate reports, Mexican authority also requires annual safety reports to be submitted to them with a table specifically in Spanish for all the events which are included in the annual safety report. And Central Ethics Committees additionally requires international safety report on a 6-monthly basis. Australia. So there is a very unique requirement for Australia, which is submission of only domestic SUSARs to TGA, Therapeutic Goods Administration, which is the regulatory authority for Australia. Timelines, however, remain the same, but TGA does not need aggregate reports to be submitted to them on a mandatory basis. So this has to be submitted only upon request. But there is Ethics Committee of Australia, which is HREC, Human Research Ethics Committee, which requires the DSUR to be submitted to them on an annual basis. So while reporting to regulatory authorities is extremely important, I think management of follow-ups is also very crucial in the safety management of global clinical trials. Regulatory authorities give a lot of weightage on how pharma companies do the follow-ups and how they report the follow-ups to the regulatory authorities. And it is said that knowledge becomes wisdom when it comes in the hands of the one who knows how to use it. So it is very important to analyze the data appropriately to give a holistic clinical picture. And so why exactly follow-up is done? So follow-ups are done to have the complete information on any missing information for appropriate assessment of the causality. The sponsor should report the relevant information after receipt as a follow-up report. Follow-ups are also done for long-term outcomes, and long-term follow-ups are done to see whether there is a resolution of the event or not. And some of the events are chronic, and actually long-term follow-ups are required to be done for such kind of events. And clock for submission of follow-up starts when follow-up information is received by the sponsor. So further, to assist sponsors in determining whether follow-up information is actually relevant to be submitted as an IND safety report to FDA, FDA recently published a guidance on when to submit follow-up reports. What all information triggers the follow-up safety reporting, and which all information does not trigger any follow-up safety reporting to the FDA. So these are some of the examples where the information, the kind of information which triggers the follow-up safety reporting, that is, in case there is a change in the diagnosis of the adverse event, or death is reported as a result of any adverse event, or we've received autopsy findings in the follow-up, or there is any other new information that actually significantly impacts or changes the assessment of the causality. There is certain other information which does not trigger the follow-up safety reporting, and some of the examples which FDA has published in the guidance is like the non-investigational treatment changes, or non-resolving adverse event updates, or any medical or treatment history that is not at all relevant for assessment of causality. So this kind of information does not trigger that follow-up information has to be reported. Though obviously, of course, such information has to be processed in the database, but it is not required to be reported to FDA as a follow-up IND safety report. So the bottom line is that any information which provides any update about change in the causality, or any information about change in the causality or medical assessment of a case, has to be reported to FDA as a follow-up. Another crucial area from SAE management perspective is how to maintain the integrity of the data in double-blinded clinical trials. Golden rule is that the investigator does not have to break the blind. They can only break the blind in some exceptional circumstances when it is necessary for the safety of the subject. So in case the blind is broken, the investigator must immediately notify the medical monitor of the sponsor, and such a patient is then not further continued in the trial. Safety team who is involved in processing and submission of safety reports unblind the safety reports prior to submission to regulatory authorities, and only reports with the study drugs are required to be submitted, and placebo reports are not required to be submitted generally, but there are certain exceptions, like for certain events like hypotension or anaphylaxis. So in this entire process of SAE or safety management, there is an important contribution of safety assessment committees nowadays. And FDA, in fact, has laid down some draft guidance as well on this safety assessment committee regarding composition of the safety assessment committee, requirements and functionalities of the safety assessment committee. What exactly is the safety assessment committee? See, it is actually very critical for sponsors to detect and report as early as possible serious and unexpected adverse reactions and clinically important increased rate of previously recognized serious adverse reactions. Timely reporting of the safety information allows FDA to consider whether any changes are required in the study conduct beyond those initiated by the sponsors themselves. For these reasons, the FDA recommends sponsors to develop a safety assessment committee for a systematic approach of safety surveillance. Now, safety assessment committee is a group of individuals chosen by the sponsor to review safety information in a development program and tasked with making a recommendation to the sponsor regarding whether the safety information must be reported in an IND safety report. The Safety Assessment Committee oversees the evolving safety profile of the investigational drug by evaluating the cumulative serious adverse events from all the trials in the development program, as well as from the other available safety information, and performing the unblinded comparisons between the investigational and the control groups. The Safety Assessment Committee thus needs to have the access in totality of the safety information from all ongoing or all completed studies, and this will contribute to the evaluation of the serious adverse events. Sponsors should have a safety surveillance plan to describe the processes and procedures for assessing the serious adverse events and other safety information. Safety Assessment Committee provides a critical oversight for analysis of the clinical trial safety events and can help to lower the over-reporting of the expected safety events to the authorities in order for them to focus more on the serious and the unexpected events. So Safety Assessment Committees are typically seen in oncology trials when a decision has to be made for any dose limiting toxicity or any dose escalation or further cohorts. So we all know that in clinical trials, there is a data monitoring committee or the independent safety monitoring board. Safety assessment committees and data monitoring committees both are comprised of medical and statistical experts that are independent of the clinical trial operations, but are responsible for reviewing the unblinded data in order to make a recommendation to the sponsor. While safety assessment committees recommend whether individual safety events should be reported as an IND safety report, data monitoring committee, on the other hand, recommends the trial level actions based on the summary data. Safety assessment committees are tasked with analyzing aggregate data across a drug development program, whereas data monitoring committee just focuses on one study. Safety Assessment Committee meet more frequently due to reporting timelines, as Safety Assessment Committee has to decide whether this event has to be reported as an IND safety report or not. But Data Monitoring Committee tends to meet at a lesser frequency, which can be quarterly or biannual. So we've been talking about SUSAR reporting with investigational medicinal product, but we also have to understand that the patients sometimes also take other medications along with the investigational medicinal product, and these are called auxiliary medicinal products. So from EU clinical trial regulation perspective, like 536/2014, so medicinal product used for the needs of the clinical trial as described in the protocol, but not as an IMP, is called auxiliary medicinal product. Auxiliary medicinal products should be related to and relevant for the design of the clinical trial, which excludes concomitant medications. Auxiliary medicinal product can be rescue medications, can be challenge agents, can be medicinal products to assess endpoints in a clinical trial, and any background treatment. So EU clinical trial regulations has laid down certain safety reporting requirements for auxiliary medicinal products. Auxiliary medicinal products can be non-authorized auxiliary medicinal products or authorized auxiliary medicinal products. For non-authorized auxiliary medicinal products, reporting of SUSARs related to non-authorized auxiliary medicinal product should be same as for the investigational medicinal product. That is, SUSARs have to be reported. These have to be reported in EVCTM, and any safety issues that arise from clinical trials should be addressed in the DSUR. For authorized auxiliary medicinal product, the safety reporting should be done in accordance with Directive 83/EC or GVP Module 6, and it states that relevant safety information should be presented in annual safety report or the DSUR of the IMP, and no separate annual safety report is required for authorized auxiliary medicinal product. So essentially, we've seen that there are so many stakeholders in clinical trial safety management, and with so many stakeholders, challenges are bound to happen. So first and the foremost challenge in clinical safety across the globe are the linguistic differences. Certain countries still require the translation of SUSARs into local language, for example, Japan and China, though English version is still acceptable nowadays in most of the other countries. With regards to the expedited reporting requirements, there are still some countries where there are specific nuances like TGA, which we talked about, that just needs local reports to be submitted. China, on the other hand, needs serious adverse reactions to be submitted to them and not just SUSARs. And if we see Mexico, so that needs more information apart from just SUSARs. Coming to the format of submission, though SIOMS is commonly used format or electronic submission is the commonly used formats nowadays, but some countries still have some country-specific forms. Like South Africa has country-specific form, India has a country-specific form in Appendix 11 and Schedule Y, and mode is electronic in some countries, but is still manual in most of the Asian countries. So they still have to be reported in a hard copy via courier, and in some of the European countries as well. And for aggregate reports, DSURs are acceptable, but still some countries have country-specific formats to be reported to them. So regulatory submissions, or we should say safety management, is a very complex process. Only key to success is to have a robust pharmacovigilance system in place, and countries are actually trying to develop some new regulations, some new guidelines to overcome some of these challenges so that safety management process can be streamlined in future. So this is all what I had for today, and I'll now open the floor for questions. Please feel free to post your questions. We will try to answer as many questions as we can, and rest we can answer via email.
Sure. Mugdha, I think in the interest of time, we'll take a few questions. I think I can see a question on the AOSC. It says, is AOSC required for all the SUSARs? When is it required to be drafted? Yeah, so first of all, this requirement for analysis of similar event is specific to the US FDA and maybe for some specific countries. Not all regulatory authorities like EMA or any regulatory authority in Europe, they don't need AOSC. But yes, as far as the FDA regulations are concerned, if there is any 7-day alert report or if there is a 15-day alert report, so the analysis of similar report has to be appended along with the MedWatch report. Of course, the MedWatch report has to be sent in the ECDD format to the FDA. But within that MedWatch report, we can always include a section of analysis of similar report, or maybe we can append that separately. So both options are absolutely acceptable to the US FDA. And as far as when is it required to be drafted, of course, as I mentioned, we do it with the 7-day or the 15-day alert report. And whenever we are even providing a follow-up information to the US FDA for any 7-day or 15-day alert report, so if there is any further update in the AOSC, so that is also required to be attached in the report along with that. I can see another question. It says, are there any standard parameters for SE reconciliation? Yeah, so as far as the reconciliation between the clinical database and safety database is concerned, there is no regulatory definition of SE reconciliation. At the end of the day, the regulatory authorities expect that the companies should have a standard process in place so that they are ensuring that your safety database and the clinical database have a consistent information for the SE. And the reason is that SE is a sensitive piece of information. So there are some important parameters like the start date of the SE or stop date of the SE or action taken or severity or outcome or causality, or maybe as simple as verbatim term. It's very important that your verbatim is same in both the safety and clinical database, and then the coding, the preferred term is coded same, or the hierarchy of METRA like HLT, HLGT. Once we have the same PT anyway, the hierarchy will be by and large the same. So it's very important that the information with regards to the SAE is consistent across both the databases, and therefore all the companies need to have a standard process of SAE reconciliation. I'm like, you don't need to do the reconciliation for CONMED or maybe the other medical history part. It's more important that you do the reconciliation of the important parameters of the SAE, as I mentioned.
So yes, Sanjeev, I can take the other question. So there is a specific question on the MHRA reporting requirement. So the question is, how are the DSURs required to be reported to MHRA, and is there any change in the requirement in the modality of submission post-Brexit? Yeah, so this is a very good question. In fact, pre-Brexit, the reports were submitted, even if you talk about SUSARs, they were submitted by EVCTM. Now the process is entirely changed after starting 1st January 2021. So MHRA has published that there can be 3 modalities, like it can be eSUSAR portal, it can be ICSR submission portal or direct gateway through MHRA submission portal. So SUSARs have to be reported via these portals. And if we talk about DSUR, so there is a specific MHRA submission portal, which sponsors also should be having the access it in case the reporting is outsourced to the CRO. So they should be giving the access to the CRO for the MHRA submissions from MHRA submission portals for the DSURs. So yes, reporting requirements or reporting modalities have changed, in fact, after Brexit, and they have to be, sponsors have to be registered appropriately on the portals for doing the submissions. And I can take another question, and it is about how do we decide whether follow-up of SUSARs require expedited reporting? Yeah, so in fact, this was discussed in the slides as well. If we see that FDA guidance has, FDA specifically states, so which all information triggers the follow-ups submissions, because they do not want over-reporting of the SUSARs to them. So in case there is any change in the diagnosis or any change in the any information which gives which makes about a change in the medical assessment or the clinical assessment of the case, if there is any information which leads to the change in the assessment, like if death has been reported or there is an important event outcome which has been reported, so that these are all the information which trigger the follow-up reports and which should be reported. And there are other events like, other information like any non-investigational changes or any adverse events which are continuing to be like unresolved, and there is no change in that. So these kind of changes or any information which actually is not leading to any change in the medical assessment or the clinical assessment of the case. So these changes should not be reported to the FDA. They should just be processed. They should not be reported as a separate follow-up report.
Sure, I'll take, I think, another question. What is the significance of Temple's corollary? So yeah, so while doing the medical monitoring, whenever we have a drug for which we are suspecting some kind of drug-induced liver injury potential, so we have to follow this particular Temple's corollary range or maybe Hy's laws. When we have a normal alkaline phosphatase, but we have some patients with AST or ALT more than 3 times the upper limit of normal, so they are classified as Temple's corollary. It's a sign of a liver damage, but within this range, you don't have bilirubin, which is more than 2 mg%. But if you have a severe hepatocellular damage, like in the highest loss, which is positive, so then you have patients with not only AST or ALT more than 3 times the upper limit of normal, but in the highest loss, the difference is your bilirubin will also be more than 2 times the upper limit of normal in those patients. But alkaline phosphatase will still be normal or will not be like 2 or 3 times the upper limit of normal, which primarily indicates that it's not an obstructive pattern. It's a more of a hepatocellular damage. I think another question is there any gold standard for doing causality assessment? Well, as I mentioned earlier, practically in the clinical trials, we can always define what is going to be related and what is going to be not related, because at the end of the day, when we are reporting any SUSAR, we have to take a decision whether it is related or not. It's a all or none phenomena. Sometimes some academic centers use some scales like Niranjo or other things, but like the WHO classification, which gives some parameters, most of the industry personnel say they sometimes use those parameters. But as such, there is no hard and fast rule. The more important thing is in the protocol, the definition of related and not related, and what comes under the bucket of related and not related is to be documented appropriately, so that the decision on SUSAR reporting can be done appropriately. I think in the interest of time, probably we can take one more question. I think you can take on this question. It says, is there any standard requirement for SUSAR reporting in EU? I know you explained about that, but probably you can mention about those details in EU for SUSAR reporting.
Yeah, sure. Thanks, Sanjeev. Yeah, sure. I'll take this question. So, in case of European countries, so timelines are standard for reporting, but obviously there are certain mode of transmission of the reports to the regulatory authorities or to the ethics committees, which vary country to country. So EVCTM generally is used as a mode of transmission for EMA or for certain other countries like Germany, Spain, Netherlands, Austria. But there are still certain countries which expect the IND safety reports to be submitted to them via email, like Poland. And if we see ethics committees, again, there are different modes of submission across Europe. So there are emails, there are portals, and some ethics committees do even expect hard copies to be submitted to them via courier. So like Poland or Bulgaria, they accept the IND safety reports via emails. Austria, Netherlands. So Netherlands has a portal, CMO portal, where the reports have to be submitted. And certain other ethics committees still need hard copies to be couriered to them, like Germany. And for certain countries, aggregate reports like the DSURs, they have to be submitted via portal, like Spain has to be submitted via ECM portal. So I mean, what I'm trying to say is that although the timelines are standardized, but with regards to the mode of submission, so they still vary across Europe, country to country. Like it can be email, it can be portal, it can be hard copy. So we have to have all the regulatory intelligence at our end to just do the submissions to the regulatory authorities or to the ECs when we are doing the safety management of the clinical trials. I think in the interest of time, we are just able to answer these many questions as of now. So please feel free to write back to us and ask your questions, and we will be happy to respond back to you via email. And last but not the least, thank you so much, everyone, to take out the time to attend our webinar. And happy Friday, and we will see you soon with another upcoming webinar. Thank you. Thank you so much, everyone. Thank you.